Imagine you are working from home, right? You're right in the middle of this massive, highly complex project.
Oh, you've all been there.
Right. And suddenly your internet just completely goes down.
The worst feeling.
Truly. So you look at the window and you realize a squirrel just chewed through the thick fiber optic cable on the telephone pole down the street.
Wow. Okay, nature strikes.
Exactly. Now, think about what your computer does in that exact moment. Does it, keep trying to frantically download giant files, overheating its processors, and eventually crashing?
No, I mean, hopefully not.
Right, no. A smart, well-designed system pauses. It shuts down all of its heavy background processes, goes into this quiet standby mode, and basically just waits for the repair crew to fix the cable outside.
It's self-preservation.
Exactly. You might look at the blank screen and think the computer's completely broken, but it is actually protecting itself. And today, we are exploring a absolutely groundbreaking 2026 Nature paper that proves for the very first time that your brain does the exact same thing.
It really is a stunning piece of research. I mean, what we're looking at here is a complete dismantling of our old understanding.
It's huge.
It is, because for decades, the way we viewed brain injuries and devastating neurodegenerative diseases, multiple sclerosis or Alzheimer's, it was fundamentally flawed. We are literally rewriting the textbook with this one.
And that is exactly our mission for this deep dive with you today. We are going to explore how tiny nicks in the brain's distant wiring, what neurologists call focal white matter lesions, are secretly controlling the health, the survival, and the decline of the brain's main processing centers.
Even when those processing centers are miles down the biological road.
Right. So to really grasp the magnitude of this discovery, let's set the virtual stage for you.
Yeah, I want you to imagine a highly detailed, glowing 3D map of a neural circuit just floating right in front of you.
Okay, I'm picturing it.
Good. So in this map, you have two main distinct components. First, you have the gray matter. These are the neuron cell bodies, the synapses.
The massive computing servers of your brain, basically.
Right. Exactly. The servers where all the thinking and processing actually happens. Then stretching far away from them, you have the white matter.
The cables.
Yep. These are the long, fatty, myelin-coated cables that transmit the electrical signals from one gray matter server to another.
Got it.
Now, historically, medical science thought that a physical lesion, like a microscopic wound in those white matter cables, and inflammation happening in the gray matter servers were two completely independent, unrelated problems.
Which is wild to think about now. It was like saying if the server room in New York caught on fire, we definitely wouldn't blame a frayed piece of underground cable way out in Ohio.
Right. They treated the two events as totally separate diseases.
But this new paper proves they are intimately, inextricably connected. So how did they prove this? Because, mapping a live brain's wiring is incredibly difficult. It's just a tangled mess in there.
It's a massive plate of spaghetti.
Exactly. So how on earth did researchers isolate a single cable without accidentally hitting the server?
That was the genius of their experimental design. They chose to look at a very specific neural pathway in rats called the olivocerebellar circuit.
Okay, let's unpack this. Out of all the places in the brain, why this specific circuit?
Because it has this beautiful anatomical separation. In this circuit, the computing servers, the gray matter cell bodies, are located deep down in the brainstem. But their long transmitting cables travel far away through a completely different isolated bundle.
So they are physically distinct.
Very. In a rat's tiny brain, these two areas are separated by a massive 3.5 millimeters.
Which, just to put that in perspective for you, 3.5 millimeters in rat brain geography is an absolute cross-country road trip.
Oh, yeah. It is a vast physical distance on a microscopic scale.
It's huge.
And it gave the researchers the perfect physical buffer to test their theory. They wanted to intentionally damage the cable out in the middle of nowhere without accidentally causing blunt force trauma to the server itself.
Right, they needed a clean test.
So to do this, they injected a highly specific fat-dissolving chemical directly into that distant white matter highway.
And we should probably carefully detail how this chemical works, because it's not just like generic brain poison.
No, not at all. Ethidium bromide, or ETBR, it specifically targets and strips away the myelin.
Myelin being that protective fatty insulation wrapped around the nerve wire, the stuff that helps the electrical signals travel fast.
Exactly. This chemical dissolves the insulation, but, and this is a crucial detail, it does not kill the underlying axon wire itself.
Which makes it the perfect biological model for a demyelinating disease like multiple sclerosis. The insulation is gone, but the copper wire is still intact.
Spot on.
Okay, wait, hold on. I'm stuck on something right here.
Yeah.
If the underlying wire wasn't actually severed, like it's just stripped of its insulation, how does the cell body, sitting 3.5 millimeters away, even know there's a problem? The physical connection isn't actually broken.
That is the perfect question. And it gets right to the heart of how neurons communicate internally. It's a process called retrograde signaling.
Retrograde signaling. Okay, how does that work?
Well, when the myelin is stripped away, the bare wire becomes incredibly stressed. It suddenly takes way more energy to push an electrical signal through an uninsulated wire.
Oh, sure, it's like a leaky hose.
Right. So to survive, the stressed part of the cable synthesizes specific distressed proteins. And then it ships these proteins backwards, like a microscopic freight train traveling in reverse, all the way up the axon, back to the main cell body.
To essentially sound the alarm.
Exactly, to say, hey, we have a major problem down here.
Wow. So the distress signal travels all the way back to the main server, and I guess this is where the upstream ripple effect becomes visible.
Yes, because the researchers then looked back at the gray matter, miles away from the injury, to see what those neuron cell bodies were doing in response.
And they used a brilliant microscopic tool to do it, right?
They did. They injected a special tracker, a calcium indicator, called GCAM-MP7, that physically flashes fluorescent green every single time a neuron fires an electrical pulse.
That is so cool. It literally let them watch live brain activity in real time.
And what they saw was shocking. By day 7 after the distant cable injury, they saw a massive 60% drop in neuronal activity in the gray matter.
60%. And keep in mind, these cell bodies were completely untouched by the chemical. They weren't physically damaged at all.
Not even a scratch. They just went completely silent.
Like the computer shutting down its background processes?
Precisely. But the silence wasn't the only thing happening. That silence was quickly accompanied by an incredible, highly targeted swarm.
A swarm of what?
Well, the brain has its own dedicated immune cleanup crew called microglia.
Right.
Normally, these tiny cells just peacefully patrol the brain tissue looking for debris. But by day 13, the density of microglia in that distant, silent gray matter absolutely skyrocketed, the threefold increase.
A total swarm. But what's crucial for you to remember here is what did not happen.
Yes, the astrocytes.
Exactly. The brain has another type of support cell called an astrocyte. When a brain suffers a generic blunt force trauma like a severe concussion, astrocytes panic and rush in to form thick biological scars.
But in this experiment, the astrocytes did absolutely nothing. Their baseline levels didn't change at all.
Which tells the researchers a major secret. This wasn't a panicked response to trauma. This was a highly orchestrated, highly specific biological program.
Highly specific and, frankly, a bit of a microscopic horror story.
Oh, wow.
Because these swarming microglia weren't just hanging around the silent neurons to offer moral support. They were actively eating the excitatory synapses of these silenced neurons.
Oh, wait. The brain's immune system is eating its own healthy connections. That sounds like a catastrophic autoimmune failure. How did they even prove that was happening?
By looking inside the cellular stomachs of the microglia.
Gross, but fascinating.
I know, right? Using special markers, they highlighted these microscopic stomachs called lysosomes and found they were massively enlarged.
Yeah.
And physically stuffed full of the exact proteins that make up the neuron's excitatory synapses.
Oh, wow. They were quite literally chewing the communication ports off the servers.
Yeah, eating them alive.
That is terrifying. But they aren't just eating blindly, right? I mean, they are following orders.
Yes. They are directed by a very specific targeting system. The researchers discovered that the gray matter synapses were suddenly coated in a specific complement protein called C1q.
And what does C1q do?
Normally, complement proteins are used by the immune system to tag dead bacteria for destruction.
Yeah.
It acts like a microscopic fluorescent sticky note that just says, eat me.
So let me get this straight. The distant cable injury triggered the distress signal.
Yep.
The distressed neuron shut down its power and then deliberately tagged its own communication ports with eat me stickers, inviting the immune system to come and devour them.
You've got it. That's exactly the mechanism.
Okay, before we move on to the twist, let's just pause. I want to make sure this highly complex biological cascade is perfectly clear in your mind because it is the foundation for everything else we are going to discuss today.
It's a lot to take in.
It is. So let's do a quick recap using a memory trigger. Let's think of the three: Mrs. Myelin, mute, and microglia.
I like that.
Right. So step 1, the myelin insulation on the distant white matter cable is damaged. Step 2, a week later, the gray matter neuron mutes its self-activity, drops by 60%. Step 3. 2 weeks later, the microglia swarm the gray matter and start eating the neuron synapses.
Terrific summary.
So, to ask you, the listener, a direct question, if your brain's immune system is actively eating your synapses because of a tiny cable nick miles away, doesn't that sound like a totally irreversible disaster?
It sounds like the very definition of permanent neurodegeneration.
Yeah.
But what is truly fascinating here, and this is the crux of the paper, is that it's not. This terrifying damage in the gray matter is actually a brilliant temporary repair mode.
It is a feature of the brain, not a bug.
Exactly. The ultimate plot twist. Because we have to look at what happens on day 28 of the experiment.
Okay, what happens on day 28?
In this specific rat model, by day 28, the distant white matter cable has successfully regenerated its fatty myelin. It's a natural healing process called remyelination.
The insulation grows back.
Yes. And the very moment that cable is fully re-insulated, the gray matter completely goes back to normal.
It's almost unbelievable when you think about it. The microglia pack up and leave the area. The synapses literally regrow from scratch.
And the fluorescent green tracking shows the neuronal activity firing right back up to its normal pre-lesion levels.
Wild. Now, to understand how the neuron actually survives this intense ordeal without just dying off, the researchers used a cutting-edge technique, right?
They did. It's called spatial transcriptomics, specifically a method called D/T-Sec.
I absolutely love this technology. Let's explain it simply because it is revolutionizing biology.
Go for it.
Usually, if you want to know what genes are active in a tissue piece, you basically have to blend it all up into a smoothie and test the liquid, but you lose all the geography doing that.
Right, you know it's there, but not where it came from.
Exactly. Spatial transcriptomics allows you to take a pristine slice of tissue and lay a grid of microscopic barcodes over it. It doesn't just tell you what genes are turned on, it tells you exactly where on the map those genes are active.
It is like reading the genetic mail of individual cells in their exact zip code without even opening their front door.
That is a fantastic analogy, and when they read the genetic mail of those silent neurons, they found something remarkable.
They really did. They didn't find any cell death or apoptosis genes. The neurons were not dying.
So what were they doing?
They were heavily expressing genes for oxidative phosphorylation, meaning they were revving up their internal power plants, their mitochondria, generating huge amounts of energy for survival and adaptation. They were also expressing specific genes that actively suppress incoming signals.
So, to use another analogy, let's look at this like a massive manufacturing factory.
Okay.
A pipe bursts in the basement. That's the distant white matter lesion. The factory manager doesn't panic and burn the building down. Instead, they temporarily shut down the main assembly line. That's the neuron muting its activity.
Right.
They fire the shipping and receiving contractors. That's the microglia eating the synaptic connections, so they can divert all the factory's electricity to fixing that broken pipe.
They are rerouting every available resource to survive the crisis.
Exactly. And once the pipe is fixed, they rehire the contractors, turn the power back on, and business resumes as usual.
A perfect way to visualize the mechanism. It is a profound example of regenerative plasticity. And we really must emphasize to the listener here, this is not just some quirky anomaly that only happens in a highly specific rat circuit.
Right. The generalizability of this paper is massive. It applies everywhere.
They replicated this exact same mechanism in mice. They proved it happens in completely different brain regions, like the corpus callosum and the hippocampus.
Right. And the hippocampus is vital for memory and learning.
Exactly. They even used entirely different methods to cause the initial cable damage. For instance, they used a chemical called lysolecithin and another one called LNIO that temporarily restricts blood vessels, essentially mimicking a microscopic stroke.
Which, let's be real, is an incredibly common event in aging human brains. Most of us will have tiny microstrokes as we age and never even know it.
Absolutely. And regardless of how the white matter was damaged, whether by a fat-dissolving chemical or a microstroke, whether in a rat or a mouse, whether in the brainstem or the cortex, the result was identical.
The distant gray matter shut down, the immune cells ate the synapses, and then everything healed and turned back on once the myelin was fixed.
It's a universal brain mechanism.
Okay, let's do another quick check-in to cement what we've learned so far. We have established a new biological rule. White matter damage triggers a temporary protective gray matter shutdown. We've seen that the genetic barcoding proves it's an active survival state, not cell death. And we know it is a universal mechanism across species and injury types.
So far, so good.
But this brings us to the most crucial pivot of our deep dive. The real, so what for human health? Because if this is a natural, beautiful, highly orchestrated healing process, Why do people with multiple sclerosis or Alzheimer's suffer permanent cognitive decline?
Yeah.
Why do they have chronic inflammation? Why doesn't their factory ever turn back on?
That is the multi-billion-dollar question in neurology right now. And the researchers answered it with one final, elegant experiment.
Okay, what did they do?
Well, they hypothesized that the gray matter only heals if the white matter actually manages to send an all-clear signal. So to test this, they decided to intentionally break the repair process.
Break it. How did they do that?
Yeah.
You can't just ask a stem cell to stop working.
You can if you use a specific chemical compound called EDU. It acts like a red light for cell division.
Yeah.
They injected this EDU compound to specifically stop OPC's oligodendrocyte precursor cells from dividing and multiplying.
Let's break that down for a second. These precursor cells are the biological repair crew. They are the cells that travel to the injury, grow up, and manufacture the new fatty insulation.
Exactly.
So by pumping this red light chemical into the white matter lesion, you are basically putting up a permanent roadblock. The repair crew can never arrive at the site. The cable stays bare and exposed indefinitely.
Correct. And the results of blocking that repair crew were absolutely devastating to the brain. Because the white matter cable never healed, the gray matter microglia miles away never got the signal to stand down.
Oh, man. The temporary factory shutdown became a permanent closure?
Exactly. At day 28, when the normal, healthy brain would have completely recovered and turned back on, the brain with the blocked repair crew was trapped in a nightmare.
What did it look like?
The microglia were still swarming in massive numbers. The synapses were still permanently tagged with those C1 key eat me sticky notes. The immune cells were trapped in an endless destructive loop of chronically eating synapses that the neuron was desperately trying to regrow.
This is so profound because when neurologists look at an MRI or a tissue sample of a brain with progressive multiple sclerosis or a brain with advanced Alzheimer's disease, what do they see?
They see chronic microglial swarming.
Right. They see relentless, low-grade gray matter inflammation. They see synapses permanently disappearing, which leads to memory loss and cognitive decline.
For decades, the medical community has been looking at that gray matter inflammation in MS and Alzheimer's and trying to treat it as the primary disease. Billions of dollars have been spent trying to develop immunosuppressants to force the microglia in the gray matter to just calm down.
But this paper implies something revolutionary. It implies that the gray matter inflammation isn't the primary disease at all.
No, it's just a symptom. Yes, it is simply the brain's natural repair system stuck in a permanent holding pattern because of unhealed microlesions in the white matter.
That makes so much sense.
Right. The microglia in an Alzheimer's patient might be doing exactly what they evolved to do. They're just endlessly waiting for a repair signal from the cables that never comes.
So if we want to stop the cognitive decline, if we want to save the gray matter servers, we actually have to fix the white matter cables. We have to figure out how to jumpstart myelin regeneration.
Precisely. It completely flips the therapeutic target upside down. Instead of aggressive immunosuppressants to quiet the gray matter, we need regenerative therapies focused entirely on healing the white matter.
Okay, let's wrap this all together. We have gone on an incredible paradigm-shifting journey today. We started with the old rigid dogma that white matter cables and gray matter computing servers were independent systems.
And we dismantled that entirely.
We really did. We explored the mechanisms, the retrograde distress signals traveling miles down the microscopic road, proving that a tiny nick in a cable sends a chemical alarm that shuts down the neuron, calls in the immune system to eat the synapses, and puts the whole system into a protective hibernation.
And most importantly, we learned that if that white matter cable doesn't heal, that temporary hibernation becomes the chronic, destructive inflammation that actually defines neurodegenerative disease.
It reveals a level of regenerative plasticity, a deep, intrinsic coupling between white and gray matter that is truly awe-inspiring.
It really is.
And to bring this directly back to you, the listener, as you go about your day, think about your own brain right now. It is an unimaginably connected system. A tiny, imperceptible problem in one microscopic area dictates the chemical and physical reality of an area miles away.
It's all one web.
Exactly. You aren't a collection of separate brain regions. You are a single, highly reactive ecosystem. Every part is waiting on a signal from another.
And understanding that deep interconnection leads to a very important and perhaps slightly unnerving realization about our own daily lives.
Right. And I want to hand it over to you to deliver the provocative thought we want to leave the listener with today, something for them to mull over.
Thank you. So consider this. We know from a vast body of other medical research that simple aging, poor cardiovascular health, high blood pressure, and even chronic sleep deprivation can cause tiny, silent microlesions in our white matter.
It's very similar to that microstroke model we discussed earlier.
Yes, you don't feel them happen, you don't get a headache,
Yeah.
so they are there,
Yeah.
accumulating over decades.
Yeah.
So the question we must ask ourselves is this. If unhealed white matter lesions cause chronic synapse-eating immune attacks in our gray matter, could our daily lifestyle choices silently be keeping our brain's immune system locked in a permanent, destructive state of false repair?
Wow.
Every time we sacrifice sleep or ignore our heart health, are we inadvertently firing our own cognitive factory workers, putting our brain into a permanent shutdown mode?
That is a question that completely changes how we view brain health and every choice we make. Thank you so much for joining us on this deep dive. Go take care of your white matter, get some sleep, keep that cardiovascular system healthy, and we will catch you on the next one.